Hello and welcome back. We are on our last video of the chapter 17 material covering hearing. So I will be talking a little bit more about some of the anatomy of the cochlea because we didn't dive into that as deeply in the ear anatomy video, but that's good because it'll kind of be fresh when we're ready to cover the physiology when we have the cochlear anatomy right there, fresh in our brains. So here we have kind of a different perspective of our inner ear, because here is our vestibular complex, everything we just got done taking a look at, kind of on the opposite side. And then the vestibular branches there. And then here is our cochlear. Cochlea kind of means snail-like. So it kind of looks like a snail's shell. That's a good name, anatomical name for it. So the cochlea has the same bony labyrinth and membranous labyrinth that we saw in the vestibular complex. It's a little bit more modified though. So if we take a look, there's a lot of stuff going on in this picture. So hopefully I'm not going to completely destroy it with all of my sketches. So this is one tube, one slice. In this one slice, we can identify the bony labyrinth, the membranous labyrinth, and where we're going to find the perilymph and the endolymph. So here, this little triangle piece here, within that is the membranous labyrinth. And within that, the blue, this is going to be our endolymph. And in these spaces, this is our perilymph. Okay, so there's three tubes, and they're actually called ducts. So we have the tympanic duct is on the bottom. We have a cochlear duct in the middle, that is the membranous labyrinth, as we call the cochlear duct. It contains the endolymph. And then we have the vestibular duct. Now, you see it says scala vestibuli and scala tympani, and then cochlear duct. You can call them all ducts, that's fine. I think your lab manual calls them that. So the vestibular duct is on the top, cochlear duct is in the middle, tympanic duct is on the bottom. But where we see kind of this repeating pattern of these three tubes, that does not mean we have 1, 2, 3, 4, 5 sets of these three tubes. It is a single set of those three ducts. So if we were to stretch and unwind our cochlea, we would have on the top, we have the vestibular duct, cochlear duct in the middle, tympanic duct on the bottom. It just happens to be curled up into a cochlea. And I know my drawings are horrible, but I do what I can, either to make it better or to confuse you. So when we take a look at this slice through, please remember that what you're seeing is a two-dimensional representation of a three-dimensional structure. And that's kind of what that's supposed to be showing. And those arrows, it's a continuous tube. So the purple is showing the vestibular duct, the orange is showing the tympanic duct, and the blue arrows are showing the cochlear duct. It is a continuous duct from the oval window all the way around to the top, and it comes back down to the round window, right? So here's the oval window, which is connected to the stapes, and here's a round window, which is just a flexible membrane. We'll see this in lab as well, and we do have a slide, so you'll get to see a microscopic slide of a cochlea where you'll be able to identify those three things. So here's our set, maybe I'll do it in a different color. Here's our set of three tubes. Here would be our cochlear duct, here's a vestibular duct, here's a tympanic duct. Okay? We have perilymph in the vestibular and tympanic duct, and we have endolymph in the cochlear duct. Okay, so let's take a closer look at what's called the spiral organ, or in older editions, it might still be called the organ of Corti, which was named after a person's name. So again, we can identify our set of three. Remember, it's this continuous set of three that goes all the way around to the top. So here's our tympanic duct on the bottom, here's our cochlear duct in the middle, and our vestibular duct on the top, right? That's going to be this repeating pattern because it's the same tube that's continuous all the way around. So some other structures that we can see here, we can zoom in a little bit closer. First of all, before we do the zoom in, I'm going to talk about the vestibular membrane, which is this thin membrane that separates the vestibular duct from the cochlear duct. And now we're going to zoom in here. So we have another membrane, which is the floor, if you will, of this spiral organ. It is called the basilar membrane. Okay, so that's where all these hair cells and supporting cells are going to rest upon. And then sitting just overlying those hair cells, we have what's called the tectorial membrane. So we have three major membranes that you need to make sure you are clear with. The vestibular membrane separates vestibular duct from cochlear duct. The basilar membrane is the floor of the spiral organ, and the tectorial membrane sits just over that. So we can see that on this micrograph. Here's our basilar membrane, here's our vestibular membrane, and here's our tectorial membrane. It's kind of curled up in this preparation. And then these are all the hair cells. These are the cells that are going to be those are the hearing part, right? So the sensation that we had to pick up for equilibrium was the pull of gravity and linear acceleration and position in three-dimensional space. What is a stimulus? So we need to be able to convert from that particular stimulus into action potentials. Well, it's sound waves. Sound waves are pressure waves that move through air and liquid. How are we going to be able to convert that stimulus into action potentials? That is why we have this very precise and special structure to be able to do that. And it works really well. The human hearing abilities are just amazing. And there's even animals in the animal kingdom that are even more better than human hearing. All right, so that's kind of the anatomy of this spiral organ. I did want to mention before we move on, we have our hair cells, okay? We have these rows of cells, and I know it just looks like there's 4 cells, but again, remember, because this is continuous, we have 4 continual rows of these hair cells that are all sitting underneath this tectorial membrane along the entire length of this spiral set of tubes. These hair cells do not have a kinocilium. They're just a regular, all of the cilia are the same length because there's no directionality to hearing. We needed directionality for equilibrium, right? Because we need to know which direction we're bending. But in hearing, it's just going to be distortion of those hair cells getting pushed up into the tectorial membrane to show that a sound wave has happened, okay? Okay, so just like we had to do a little physics lesson on light and bending of light in our lens discussion, we have to have a little discussion of sound, a little bit of physics discussion of sound before we can apply that to the physiology of hearing. So sound is a pressure wave as opposed to a, now I just lost all my kinds of waves, like an orbital wave, like in the ocean. That's a different kind of wave. This is, when you make a noise, right? That sound, the vibration of my hands hitting together is kind of compressing air molecules together. That is our sound wave, this pressure wave. And so here's the air molecules. We can see that here with the tuning fork. You whack the tuning fork, it's vibrating the air molecules. That sound wave then is going to be funneled into our ear and eventually onto our tympanic membrane. And it's a tympanic membrane that's going to vibrate at the same wavelength as the tone that's making the sound, whether it's tuning fork or my voice or your cell phone or clapping or somebody else talking, whatever the sounds are, they all are going to have their own specific wavelength. So again, wavelength, we saw wavelengths with light. Here's wavelengths also. The loudness of the sound is amplitude. So if you have a quiet sound, you might have waves like this. A medium sound, loud sound, all at the same wavelength, which would be the same kind of tone or pitch. Amplitude gives you volume. How loud is that sound? When we talk about sound and health or potential damage to your hearing, or measuring the amplitude of sound, we can have the decibel levels and the decibel scale. And I do believe the decibel is exponential in nature. So you have an exponential increase in the intensity of the sound as you go up per unit. So it starts at 0 is the lowest audible sound for humans. Human decibels are a human invention or a human application. So if you could imagine a quiet library or a soft whisper, that would be around 30. And here we have our threshold of damage. So up to about 60 decibels, which is an air conditioner at 20 feet, a conversation, sewing machine in operation, you can have extended exposure to that level of sound and not have any potential damage to your hearing structures. And it could be any of the structures, the Eardrum, the bones, the oval window, the round window, the basilar membrane, the tectorial membrane, or the hair cells themselves. Any of those could be targets for damage that would lead to hearing loss. Busy traffic and noisy restaurants, some damage if continuous. Subway, heavy city traffic alarm, clock at 2 feet away, factory noise for more than 8 hours. That's an 80 decibel level. That could be dangerous and potentially cause damage. So it gets, immediate danger. Heavy metal rock band. I have to admit, I have been to a few of those without earplugs. So that's all the way up to 120 decibels. You could potentially have hearing damage happen. Rocket launching pad. People are not allowed on rocket launching pads, but if you were, you're going to have hearing loss. That's why people aren't allowed, other than the huge blast of flame and heat that come out of a rocket engine. All right, so let's start this hearing process. What's going on? So we just discussed that sound is a pressure wave. So here's our pressure waves coming in. And because our tympanic membrane is flexible, it's going to start going like this. And it's going to do this in resonance, which means as the sound waves are coming in, the tympanic membrane is going to vibrate at the same wavelength, if you will. So then that vibrating tympanic membrane will then vibrate our 3 ossicles. Their job is to amplify this pressure wave. So now we've gone from a pressure wave in the air to a mechanical movement back to a pressure wave at the oval window. The oval window, this flexible membrane, is connecting to the vestibular duct, right? So this is the vestibular duct is on top. And now we have an air wave converted to a mechanical wave, which is like a little hydraulic pump that's moving back and forth, creating a hydraulic pressure wave, this fluid pressure wave in the vestibular duct. Okay, so that's what's happening, kind of steps 1, 2, 3. Okay, so that wave will continue to go around the vestibular duct, and it will continue to go around, but sometimes at a particular location in the cochlear duct, that wave will kind of transfer across the cochlea, and it vibrates the basilar membrane. Okay, so if you remember, we had our, if I'm going to draw my cochlear duct here. Okay, we had our, let's see, another one of my bad drawings. So here's our vestibular membrane, here's our basilar membrane, and so here's our vestibular duct and our tympanic duct. And if you remember, this was our cells. So we had all of our hair cells with all the little hairs, and this was our tectorial membrane sitting over the top of that. So as the pressure wave comes in, it is going to distort that basilar membrane, and it's going to push those hair cells up into the roof, the tectorial membrane, causing the cilia to distort, opening those mechanically gated channels, just like we saw in equilibrium, allowing sodium to rush in, depolarizing the cell, sending the signal to your cochlear branch that a sound has been picked up in that particular part of the cochlea. Okay, so that is basically the hearing process. The sound wave is converted into a mechanical wave, which sends a fluid pressure wave through the cochlea, distorting that basilar membrane, pushing the hair cells up into the tectorial membrane, and voila, that will be picked up by the neurons. Okay, so here's the whole process in one picture. Sound wave, mechanical wave, fluid wave, distortion of the basilar membrane, signal is sent via cochlear nerve. Now you might be asking, well, how do you know which part of the basilar membrane is going to be distorted? Well, the physical nature of the basilar membrane affects how flexible it is. So it is really dense and really thick up here close to the oval window. And as you get further out, it's a little bit less thick. And then when you get out here, it's really flexible. So the more dense this membrane is, the more energy is needed to distort it, to push those hair cells up into the tectorial membrane. So high frequency sounds with a high energy, this is 16,000 hertz, that's going to distort the membrane close to the oval window. Medium sounds, when we're over here, that will distort the membrane at that particular frequency because the membrane is more flexible there. The lower hertz with the less energy, so the 1000 hertz, this would be like a low, very low tone, then we would cause flexible, distortion of the basilar membrane way at the very end of the cochlea, which will be determined to be a low frequency sound. So remember, there are hair cells located all along the entire cochlear duct. So wherever that basilar membrane is distorted, those are the hair cells that are going to be depolarized, which are then monitored by neurons. And so we get a very precise information about what tone the sound is, a high frequency, a mid frequency, or a low frequency sound, and everywhere in between. And that is because of the thickness going down to less thick and pretty thin by the time we get to the end, because it is a physical property of the basilar membrane that affects when and where it is going to be distorted. Okay. So what's cool about that is because every one of those hair cells in that whole row along the entire spiral organ is monitored by different cells, that information then goes up to your auditory cortex, and we have, just like we can map different regions of the brain wrinkles for, the motor control, we have high frequency sounds kind of towards the insula and low frequency sounds more towards the outer surface of our temporal lobe. And that is our auditory cortex, which then can be processed by our auditory association areas. Okay, so that pathway, so the sound, the stimulation of the hair cells at the specific location of the cochlea is picked up, travel along the cochlear branch. It sits, gets relayed by the nuclei, again, cranial nerve 8. Nuclei sits in the boundary between the pons and the medulla, gets sent up to our inferior colliculus, medial geniculate nucleus, which then sends it up to our auditory cortex. So a lot of review of those brainy parts that we learned back in chapter 14. Alrighty, I think we did it. Yes, we did. So that's our special senses. We have one more chapter to cover before our second exam, which will be endocrine, which we are done with nervous system chapters, ready to move on to another system of control with chemicals. Instead of action potentials, we'll be talking about chemical messengers like hormones. So I will see you next time. Bye.